Good afternoon. My name is Jacob Huang. I'm the Executive Director of IAS of CDU. And I think it's very difficult to organize an event, especially extended workshop during the summer. So anyway, I hope the speaker don't mind. And anyway, this can be a very good exchange opportunity between all the professor and the student. So if people see the back, I invite you to move a little bit forward, so it's easier for you to ask questions. Okay. So today we are honored to invite a very famous professor, Professor James Douglas ENGEL from University of Michigan Medical School. And from my record, I think he got his bachelor degree in chemistry back in 1970. So you can sort of calculate how old he is right now. Almost 70 something? Yeah. Oh, wow. Okay. So, and also he got his PhD also in chemistry in University of Oregon. Then he completed his postdoc in molecular biology in Caltech. Okay. And that's very close to my school, UCLA. Okay. And Professor Douglas is the chair professor of developmental biology, right? And also is the chairman of cells and developmental biology, and also co-director of the Center of Organogenesis, right? And I think he got his PhD work devoted to understand the thermodynamics behavior of methylated DNA, and also as a postdoc in Caltech, he generated the first genomic DNA libraries there, and also he's the first one to show the hypersensitivity sites, right? In their quarantine, and that is coincided with the transcriptional regulatory elements. Okay, if I pronounce it wrong, forgive me. I'm trying my best. Yeah, so maybe I can invite all the biology people to name some of the material science terminology. That will be also very difficult. Yeah, but it's all new to me, very interesting. And they discussed the called enhancer, yeah, and subsequently discovered the enhancer function by looping through the chromatin to directly interact with the promoters. Okay, and then they discovered the GATA. I think probably one of the topics today, transcription factor gene family, and show that a GATA3 loss function will lead to the embryonic lethality. Okay? And they later found the conditional GATA3 loss of the function will also lead to the complete abolition of T cell lineage. Okay? And also they have done a lot of pioneer work on the BACs and YACs. I don't know what that is. Maybe you know. Okay. And also he showed a lot of interaction for the repressor and some new findings. So today, Professor ENGEL will talk about GATA3 regulates multiple aspects of. This is called thymocyte development. So let me give a big applause to Professor.
Thank you very much for a very nice introduction. The only part that I'd like to correct is I stepped down as chair of cell and developmental biology 4 years ago. So now I have a lot more fun just doing science. So thank you to Professor Smale and to Indika for inviting me to this workshop. And today, I've heard that not everyone here is a biologist, so I'm going to spend a little bit of time going through some background so that we'll all start sort of on the same page. And I'll explain during the course of the talk what BAC and YAC actually stand for, okay? They're backs and yaks. So just to get us started, I'm going to show you a slide that is very common in biology that we all know and understand and love, that hematopoiesis starts as a single cell in the bone marrow called the hematopoietic stem cell. Stem cells give rise to a variety of different progenitors and then precursors as they differentiate. And then finally, they give rise to all of the mature blood cell lineages that circulate throughout your body. As was mentioned in the introduction, our work began on T-cell biology with the discovery in 1990 that there was a group of transcription factors called the GATA factors, so named because the core motif recognized by these transcription factors is the nucleotide sequence GATA. When we first cloned this gene family, they weren't called anything, so we had to make up a name, and we called them NFE factors for nuclear factor erythroid, because that was the first gene that we went after. But subsequently, we found out that all of these factors are expressed in different cell lineages, and consequently, continuing to call them that was a misnomer. And so we decided to name them after the core nucleotide sequence that was recognized by all of the members of this family. So we showed originally in this paper that the GATA factors were expressed in a different pattern for each member of the family. So, for example, GATA1 is expressed almost exclusively in erythroid cells. GATA2 is expressed in, it turns out, a very wide variety of cells, almost all of which are in active proliferation. GATA3 is unlike GATA2 and is more like GATA1 in that it's expressed in very specific cell lineages, but unlike GATA1, it's expressed predominantly, as you can see with this blowout signal on this RNA blot, it's predominantly expressed in T cells developing thymocytes. So in this hierarchy of cells, what we've discovered, we and many other colleagues over the years, what we've discovered is that GATA3 is restricted to expression in the stem cell and multipotent progenitor compartment, as well as in T cell and T cell derived lineages, but not in B cells, the other lymphoid compartment, and not in any myeloid cell compartment. And I'll show you the evidence for that as the talk goes on. So what's been discovered over the last 20 years, or almost 30 years now, since we discovered this family of transcription factors, is that GATA factors underlie multiple human pathologies. So, for example, GATA1 mutations are a key driver in trisomy 21 associated myelodysplastic syndrome. GATA2 mutations lead to monomac or Emberger's syndrome, which affect a variety of different tissues. Inversion of GATA2, a common inversion of GATA2, leads to a specific kind of leukemia. GATA3 loss of function in one allele leads to a syndrome called HDR, which is short for hypoparathyroidism, renal dysplasia, and sensorineural deafness. I don't expect you to remember that, but we will have a quiz at the end. GATA3 is also expressed overexpression in a subset of patients with peripheral T-cell lymphoma. So these are important transcription factors in a variety of human diseases and syndromes, and we would like to understand the etiology of GATA3 function in T-cell development, because its most important role, we discovered early on, is in development of the T-cell lineage. And this is what I'm going to tell you about today. Now, a lot of the studies on GATA3 function in T-cell development have been at later stages when T-cells begin to produce active receptors and move from the thymus into the bloodstream and become activated at secondary immune tissues. But what I'm going to tell you about today is work, if I can get this to forward, all right, is work that we've been studying focused almost exclusively on the thymus. So this is the most interesting aspect of T-cell development as a developmental biologist. As an immunologist, everything that's interesting goes on out here. So T-cells begin with hematopoietic stem cells, and then as they differentiate into multipotent progenitors, they circulate out of the bone marrow into the bloodstream, and it's these theoretical cells called thymic seeding progenitors, which migrate into the thymus and become the first recognizable T cell, the early T cell progenitor. ETPs then differentiate through a series of stages called double negative, meaning that they don't express either the CD4 or CD8 co-receptors. And then finally, they become double positive. They express both. And then finally, in the thymus, they express only one of the two. Those cells then exit the thymus and proceed to populate peripheral lymphoid organs, where they become activated and fulfill their effector function in adaptive immunity. So what I'm going to tell you about today are the things that go on in here, several aspects of things that go on in here when the cells are just beginning to develop and commit to the T-cell lineage. The reason we can study T-cells in the thymus, even though there are really very few cells, we have very nice specific cell surface markers that we can use flow cytometry to distinguish between these different developmental stages. So it's very simple to analyze cells as they go through differentiation between these different stages, and we can tell if there are defects in any of those maturation steps. So questions that we set out to address regarding the function of this critical T-cell transcription factor were, first, who regulates GATA3? That is, what signaling molecules and transcription factors act upstream of GATA3 to initiate its transcription at different stages of T-cell development? Which genes does GATA3 itself regulate downstream? And finally, and probably most importantly, what does GATA3 actually do? What does it elicit inside T cells that makes them progress from one developmental stage to the next? So starting out looking at the first question, we first tried to figure out which transcription factors and upstream activators activate GATA3. And the way we set out to do that was a little unconventional, because after several years of study, we determined that sequences near the gene were not responsible for its transcriptional activation in T cells. So the sequences had to lie somewhere else, either far away or close within the locus. And so we undertook a strategy called backtraps to identify the DNA sequences that were responsible for activating GATA3 in T cells. And the way this worked was to take a series of overlapping bacterial artificial chromosomes containing sequences that were somewhere near the GATA3 locus, and then insert, using homologous recombination and recombinering, either a GFP or a LAC-Z reporter gene by homologous recombination into the vector backbone. So each back had a reporter gene inserted into sequences which were not necessary for its expression. And if there were any DNA sequences lying within that back sequence, then they should be able to fold back and activate the reporter gene in T cells. When we did that, we actually covered about 2 megabases of sequence. I've shown only the 3' megabase here, of overlapping bacterial artificial chromosomes, each of them marked with one of these reporter genes, in this case, the LAC-Z reporter. What we discovered was that there are a remarkable variety of enhancers that are located anywhere from 100,000 base pairs up to 1,000,000 base pairs away from the transcriptional start site. And these are spread throughout the locus, and this slide just shows a few of where those enhancers lie, either more or less precisely relative to the transcriptional start site. So there are 2 different kidney enhancers. There's a sympathoadrenal enhancer, a pyloric enhancer, and a boundary cap cell enhancer. So several different lineages of cells depend on GATA3 to activate gene expression in a very temporally and tissue-specific pattern. So the thing that was most interesting to us, though, was that there was 3 backs located about 300,000 base pairs, 3' to the gene, that each of them gave a very similar pattern of expression in transgenic mice that fluoresced or allowed the cell thymocytes to fluoresce. And we could show that each of these backs that overlapped by approximately 25 kb had essentially the same pattern of expression in thymocytes. So we identified a sequence here, about 300 kb, 3' to the locus, that was responsible for activation of the GATA3 gene, specifically in T cells. We went forth to study that sequence in a little greater detail, and what we found was that within that 25 kb, there was a 7 kb sequence that had very high sequence homology between mouse and human. There were a number of DNase hypersensitive sites, which are indicative of activated chromatin, and there were other signatures of transcriptional activation within this 7 kb sequence, which we then called TCE1. So Saki Omura, a graduate student, a postdoc, who has since left the lab, dissected TCE1 and determined that there was a core sequence of 1.2 kb within that larger fragment that conferred expression to a majority of transgenic mice, but if it was deleted from TCE1, there was no expression. So we believe that this 1.2 kb sequence contains the core enhancer for GATA3 in T cell expression. We then studied the sequence in greater detail and determined, using PCR and bioinformatics to identify transcription factor binding sites, that there were a variety of different transcription factors that could be shown to bind to this sequence. And we proved that those factors were indeed bound there by CHIP PCR. We then went on more recently to look at all of the transcription factors that might be binding within this core sequence, and using a TAC-seq, accessible transposase, access to transposable accessible chromatin and high throughput sequencing, that there were a variety of different transcription factors that could bind within that sequence. And we haven't tested them all yet, and nor do we plan to. But you can see that there's a lot of different transcription factors that are capable of binding within TCE1. So a key question that you always want to ask in biology is that sequence actually necessary for GATA3 T cell activation? And so using CRISPR-Cas mutagenesis in mouse eggs, we deleted the TCE1 sequence in the germline and asked whether homozygous mutants missing that sequence would express in T cells. And what we found was that, so don't pay a lot of attention to this slide. I just want you to notice that in GATA3 loss of function mutants, when we delete GATA3 entirely, there's a dearth of T cells at each of these developmental stages shown in red. But when we deleted just the TCE enhancer, we found the surprising result that this stage was affected, and this stage, and these two late stages, but none of the intermediate stages were affected. And so what this immediately told us, or led us to immediately hypothesize, was that we were missing another T cell enhancer that's required for GATA3 expression specifically in thymocytes. So the question for us immediately was, how did we miss it during that back screen? Is it possible that the sequences lie outside of the 2 million base pairs that we initially screened? Or was there another possibility? And we think that the reason that we missed the TCE2, we've come to call it, is because if you look at a typical thymus, over 98% of the cells in the thymus are mature cells and are not represented among these so-called double negative stages. So we were only looking at a very large population of double positive cells, about 85% of the cells in the thymus, when we did the initial assays, and we didn't really look at these earlier stages. So now we've gone back and we're re-assaying those same backs to look and see whether they express at these earlier stages. So as to the second question, who does GATA3 actually regulate? Why does it need to be differentially expressed at different stages of thymocyte development?
Sorry, is it possible to ask the question?
Yeah, please.
So, I understand that they are far away in the sequence.
Yes,
Could the genome be folded in such a way that they're spatially very close?
of course. So, yes, you've hit on one of my favorite themes. We showed almost 40 years ago now that the beta globin genes are activated by an enhancer looping back and actually touching the gene or being in very close proximity to it. So we think that that's how enhancers always work, and we also think that that's the basis for that backtrap assay, right? That's why it works. Okay? Any other questions? All right. So let's go on and ask who does GATA3 regulate? And the way we went about asking that question was to compare the expression of GATA3 in a mild loss of function model and compare the expression in that model to wild type cells. So we inadvertently generated a hypomorphic mutant of GATA3 by making a mutation in the germline in which the GFP gene, a reporter gene, was fused to GATA3. It should have been in frame and everything should have been normal, but it turns out that appending a GFP reporter to the 3' end of GATA3 actually decreased its stability, and so we accumulated only a much lower level in the mutant than in the wild type gene. So using a transplantation trick that I'm not going to tell you about right now, we were able to generate cells that were either wild type normally or that contained this hypomorphic mutant and perform RNA-seq looking at the levels of RNA produced in the cells that had wild type GATA3 or much lower levels of GATA3. And in so doing, we hope to identify genes that were either upregulated or downregulated with the loss of GATA3 function. And when we did so, what we found was that there were actually a very small number of genes that were directly affected. There were about 54 genes that were downregulated by greater than a factor of 2, and there were about 64 genes that were upregulated by about the same amount. And so what that meant was that the ones that were down meant that in wild-type cells, GATA3 is at higher levels than in the mutant. That means that GATA3 is required for activation of those genes in wild-type cells. So we identified this group of genes, and what do we do with that? Well, there are a couple of different ways to look at which of those genes might be important for GATA3 downstream function. The one we chose to examine was to use a candidate gene approach, and so we guessed at which of those genes might possibly be important in T-cell development. So we took 12 downregulated genes. We validated that they were in fact downregulated in the mutant cells, and that there were GATA3 binding sites somewhere within about 20 kb of that gene sequence, perhaps implying that GATA3 might act on those genes. We then undertook a strategy to identify which of those genes might be important for GATA3 in T-cell development by the following observation. In wild-type cells, we know that cells at the DN3A stage mature to DN3B and then expand explosively as they go through T-cell development. But the mutant cells undergo much less proliferation. And so the question was, if we add back any of those other genes that we identified that were downregulated, does that then rescue GATA3 expression in the later stages, right? So it's a classic rescue experiment. The way we assayed for whether or not we rescued was to include each of those 12 genes into a lentivirus or did we use it? Yeah, I think a lentivirus in this case. And then infected very early thymocytes, grew them under conditions that would allow initial expansion of the cells, and then plated those slightly expanded cells individually on feeder layers that would allow them to further expand. We followed them for about 10 days and then asked whether any of the wells contained cells marked with the lentivirus, and then we could sequence the DNA in these expanded T cells to ask which gene was able to rescue expansion into a clone. Okay? Does everyone understand the strategy? So if we infected with a virus that contained a gene that was irrelevant, it wouldn't allow expansion of that precursor. But if we infected with a gene that would allow further differentiation, we would expect those cells to be green because the lentivirus itself was green, and would allow cell expansion. Okay? When we did that, what we found, and I don't have the data to show you today, what we found was that 2 of the genes could partially rescue, and they had 2 very different phenotypes. One was a transcription factor, which has been implicated in other aspects of T cell development, which we're following up. Another was a gene called CPA3, carboxypeptidase A3. Now, I have no idea how a carboxypeptidase could rescue intermediate T cell development. So we're stuck right now with trying to figure out how to approach how that the elevated expression of that gene is important for further T cell differentiation. So if any of you have ideas or recommendations, I would love to hear them. All right, finally, what cellular processes does GATA3 actually control? This is the aspect of this study that I'm most interested in. So here's an experiment we did 10 years ago showing that GATA3 was actually absolutely required for T cell development. The way we did this experiment was to conditionally inactivate the gene, delete it, in hematopoietic stem cells, use those stem cells to repopulate mice, to repopulate the entire blood system of mice, and then ask which cell lineages are affected. What we found was that all the myeloid cells are unaffected. All of the B cells, the complementary side of the adaptive immune response, are unaffected. But there are absolutely no T cells generated in this model. And if we follow T cell development by stage, what we can see is that in hematopoietic stem cells and precursors, there's no effect, but immediately upon commitment to the T cell lineage, there's absolutely no T cell development. So GATA3 is required for all of T cell development. Now, I'm going to go into a little more detail and tell you about the GATA3 hypomorphic mutant that we made because it's important for this next section. This is a mutant, as I said, that we fused GATA3 to GFP. This was done by Kim Lim and Tagashi Moriguchi in the lab. And what we found was that if we look in thymuses at heterozygous mutants, the amount of mutant GATA3 and wild-type GATA3 differ by about tenfold, and that's quantified over here. So in mutant thymi, GATA3 mutant protein is only produced at about 8 to 10% the level of the wild-type protein, thus making it a hypomorphic mutant. So when this is an experiment, I have no idea why Tomo did this experiment, But it turns out to be the basis for a lot of the work that we've done for the last 10 years. And here's what he saw. When you follow T-cell development in a flow cytometer, you can identify the stages quite nicely using all of those cell surface markers I told you about early on. And what he observed was in a wild-type cell that GATA3 expression was normal, but in a mutant, in a heterozygous mutant, what he saw was that, shown in red here, is that there was no expression essentially early on, but then you begin to see a little bit of expression at this somewhat later stage, but by the time it gets to mature DN4 stage, the most mature double negative stage, we see abundant expression of the hypomorphic mutant. So this observation really puzzled us for a long time, I have to admit, longer than it should have. So at first what we thought was that this is some kind of artifact, right? Flow cytometry or some kind of artifact of using a hypomorphic mutant. So what we did was we compared the profile to the profile seen with a null mutant. And in this case, the null mutant was a LAC-Z knock-in. So we replaced the whole GATA3 gene and asked what happened in T-cell development when we followed the expression of this null mutant, which we could track by LAC-Z expression. And what we found was, once again, there was very little to no expression in early stage T cells, but we begin to see a little bit of expression at DN3, and then by DN4, essentially we see two populations that are quite distinct, right? And so the conclusion from these experiments was that thymocytes express neither the hypomorphic nor null mutants prior to the DN2 stage. So that started to make us believe that this phenomenon was real, and we scoured the literature to find out what might be going on. And what we finally settled on, and turns out to be the case, is that we have developmentally regulated monoallelic to biallelic gene expression. So monoallelic gene expression has been known for a long time. X chromosome inactivation is the clearest example, where one chromosome is inactivated in females so that males and females have equivalent expression of X chromosome genes. But certainly a few years later, it was shown by Shirley Tillman and many others that genomic imprinting led to parent of origin dependent gene expression. And finally, more recently, about 10 years ago, or even a little more, maybe 15 years ago now, it was shown by several investigators, almost all of whom were associated with MIT, that there's a random allelic choice of specific gene activation. And this occurs now in a variety of different systems. It's been shown to be the mechanism of activation of odorant receptors, protocadherin genes, a variety of other genes. So we suspected that what we might be observing was an example of early monoallelic expression, and then at mid-development, for some reason, half of the cells began to express both alleles. So, a very brave postdoc, Cha-Ju Ku, decided to test whether or not there was this mid-thymocyte monoallelic to biallelic switch. And there were very nice methods worked out for doing those tests. The first of them was a simple sequencing experiment. We knew that there was a silent mutation in the third exon of the GATA3 gene in C57 black 6 animals compared to FVB. So all we had to do was to breed these two mice, isolate single cells, and then ask whether the mice expressed exclusively the FVB allele, the B6 allele, or both in a sequencing ladder. That was pretty straightforward. The other way was to ask, looking at single cells, if we could detect primary transcripts of the GATA3 gene on one or both alleles. And to make a long story short, here's one of her one of her photographs. Using primary transcript RNA fish, we did an internal control of labeling with CD45, which is expressed in all hematopoietic cells. And what you can see is that in these HSC, I believe this is HSC, or ETP, there's only one signal for GATA3. And so Cha quantified GATA3 expression using both of these methods, and what she found, not surprisingly, was that in ETP and HSC, there was almost exclusively monoallelic gene expression, but then at DN2 stage and gradually increasing, we saw biallelic expression. But note that half of the cells still maintain monoallelic expression. So only about half of the cells switch. Okay? Key point. So why? So we came up with a model that we tested, and that's what I'm going to tell you about for the remainder of the talk. So the hypothesis was that in HSC and in progenitor cells in the bone marrow, cells could express either allele. It was random choice. But cells that expressed the mutant allele couldn't survive. There wasn't enough GATA3 in those cells to allow them to survive. And so only cells as they matured could begin to express the second allele. And what we were left with somehow was cells that expressed only the wild type, or cells that expressed both the wild type and mutant alleles. Okay. So if so here's the details. If we look during T cell development, if we first turn on the wild type allele in ETP, then those cells can mature and can, but don't always, express the second allele. Whereas if they start out expressing the mutant allele, they undergo apoptosis and die. All right? That's the idea. Turns out that doing all this single cell sequencing, as you can imagine, was quite tedious. We had to do 47 rounds of PCR, nested PCR, using 31 different sets, primer sets, and so that was for each cell. And we had to examine each cell independently. So we came up with a way of using barcoding in PacBio next generation sequencing to be able to examine 1,000 cells at once. And the strategy hasn't been published yet, but we hope that it will be soon. And basically what we do is use the same non-overlapping primers, amplify both possible alleles, and then using a set of primers that are specific for each V region and J region and D region, generating all possible combinations of amplification signals. And then at the very last step, we use barcodes to ensure that we are amplifying only a single sequence from a single mRNA. And then, using packed biosequencing, we can get an amplified signal from each of those sequences. And what you can see over here on the right-hand side is that if we use bone marrow cells, any nucleated bone marrow cells, we only see two bands that correspond to this sequence to here, or this sequence to here. In the bone marrow cells, but in thymocytes, we see two bands of different sizes in each one, indicating that different recombinations have taken place inside each cell. So we can very rapidly examine the recombination events in single cells in 1,000 single cells simultaneously. That's made our life a lot easier. So, what do we know so far? GATA3 is required for all of T cell development. It's monoallelically expressed at the ETP and DN2 stages that differs from all other known regulatory mechanisms. At the DN2 or 3 stage, half of the thymocytes turn on the second allele. Very curious behavior. And the mono and biallelic states in thymocytes are stably propagated. And I haven't shown you those data because it takes too long for me to explain. But it's published, and I'll tell you what the paper is if you're interested. So why might thymocytes undergo this very bizarre behavior, or unique behavior, at mid-thymopoiesis? Well, there are a bunch of things that happen right at that stage. At the so-called DN2A stage, T cells exit the cell cycle and initiate T cell receptor rearrangement. So in order to progress from the DN3A to DN3B stage, T cells need to make a T cell receptor, a functional, a potentially functional T cell receptor. If those rearranged T cell receptor genes generate an intracellular complex, the cells sense that that complex is made, and then the cells inhibit recombination of the second allele. If a successful recombination event doesn't take place, then cells go on and take a second shot by rearranging the second allele of the TCR beta gene. Okay? If the second allele rearrangement is successful, or the first, T cells happily go on and mature and undergo positive and negative selection, all the things that they normally do. But if the second allele rearrangement is unsuccessful, then the cells die by apoptosis. Okay? So there's an out. T cells can productively rearrange the first allele. If they don't, then they get a second shot. They can productively rearrange the second allele. If both of them are unproductive, if neither of the rearrangements yields a T cell receptor that's active, then the cells die. Okay? So, that process is called allelic exclusion. The decision for whether or not to turn on and recombine the second TCR beta allele is called allelic exclusion. And it's been referred to as in the literature as the lingering enigma of the allelic exclusion mechanism. This is a question that's been around for 40 years. And this occurs both in B cells, in the B cell receptor, immunoglobulin gene receptors, or T cells in the T cell receptors. So it only happens in the immune system. We don't know if immunoglobulins undergo the same kind of biallelic and monoallelic expression of another transcription factor in B cells, but certainly the mechanisms have a lot of parallels. So TCR beta allelic exclusion starts with both alleles in a germline configuration, that's I've referred to as GL, and then at the DN2 stage, both alleles undergo a D to J joining. They undergo a normal recombination event on both alleles. At the DN3A stage, cells cease proliferation. They recombine the first allele, and if that recombination event is successful, then the cells end up in a VDJ, what we call a successful or productive rearrangement, VDJ plus, or a germline allele. If the first recombination is unproductive, leading to a VDJ minus recombination event, then the cells can again undergo a second rearrangement. And again, if that is successful, those cells can then mature, whereas the VDJ minus on both alleles die by apoptosis, just what I told you. This process of allelic exclusion has been, as I said, a mystery for years and years, and the hypothesis that we are proceeding under is that in ETP cells, the first thing that happens is that GATA3 is expressed, and then they progress to the DN2 and DN3A stage by allowing recombination of the first allele of the TCR beta gene. If that's successful, then the TCR beta gene produces a productive TCR beta mRNA, which produces TCR beta protein that can then recombine with a pre-T alpha chain, and that will feed back and stop recombination of the second TCR beta allele. However, if that first recombination event makes an unproductive TCR beta protein, what happens is that the second GATA3 gene turns on. We don't know how that signaling mechanism works, but turning on the second allele leads to more abundant GATA3 expression. That then allows second allele recombination, and once again, that can either yield a productive TCR beta rearrangement or an unproductive one. These cells die, and both of these kinds of cells can go on to mature and become active T cells. Okay, so how do we test this hypothesis? So once again, if we go back to single cell sequencing of rearranged versus unrearranged germline alleles of GATA3, if we look in cells, only in cells that have to have produced an active TCR beta rearrangement, that is, a DN4 stage cell, by definition, has produced an active TCR beta protein. If we look in wild type cells, turns out that half of them express a VDJ plus allele, and then a germline configuration of the other allele, and half of the cells express a VDJ plus and a VDJ minus allele. So let's manipulate GATA3 levels and ask what happens in other cell types. So as I told you earlier, at the DN4 stage, when we should have an active TCR beta protein, we see two different peaks of GATA3 expressed in those cells. So what happens if we take the GFP plus and GFP minus cells and sequence those TCR beta alleles? The prediction of the model is that in the green cells that have higher levels of GATA3, we'll see 2 rearranged alleles, whereas in the white cells, we'll see only 1 rearranged allele, and that will be a productively rearranged allele. Okay? When we do the experiment, what we found is that in the green cells, we have the vast majority of them have 2 rearranged alleles, and some of them are VDJ plus minus, and some of them are VDJ minus minus, unexpectedly. Those cells presumably were destined for cell death, but we don't know. But more importantly, in the GATA3 mutants that only express low levels of GATA3, 90-some% of those cells have only a VDJ plus DJ configuration, meaning that they were successful on the first rearrangement. Okay? So that's entirely consistent with the hypothesis that expression of one GATA3 allele leads to successful rearrangement of the TCR beta gene the first try. But expression of 2 GATA3 alleles leads to rearrangement, full rearrangement of both VDJ genes of both TCR beta alleles, perhaps successful and perhaps unsuccessful. Okay? So in these cells, we've been able to show that that's consistent with the model in which GATA3 drives second allele rearrangement. Well, can we examine that in another way? And one of the curious things that we learned going through standard flow cytometry was that even in wild-type cells, we can see a difference in GATA3 levels by flow cytometry. So using standard GATA3 antibody, we found that in all of these mice, male or female, that they each produce 2 different peaks of low and high levels of GATA3. So let's do the same thing. Isolate cells that express low levels of GATA3 and cells that express high levels, and ask what those TCR beta genes look like. And again, not surprisingly, GATA3 low cells have 96% of the TCR beta genes in a VDJ plus DJ configuration, whereas the GATA3 high alleles have only 8% of the cells in that configuration, with both the vast majority have both TCR beta alleles rearranged. Again, entirely consistent with the hypothesis that GATA3 levels are regulating allelic exclusion. Finally, we can overexpress GATA3, in which case we would anticipate that the vast majority of cells will have rearranged both alleles once again. And when we do the experiment, that's what we find. Over 80% of the cells have either productively or unproductively rearranged both alleles. So what we believe is going on is that GATA3 abundance is controlling somehow allelic exclusion. Now we know a few more things about how it's doing so. So one of the early experiments performed in 1988 by Mike Steinmetz's group showed that transgenic expression of a TCR beta gene actually suppressed allelic exclusion in those thymocytes. And so when we tested whether or not GATA3 overexpression was regulated upstream or downstream of the TCR beta feedback loop, what we found was that it was unaffected. So that means either that GATA3 is acting upstream of the TCR feedback, or that they're acting in parallel pathways. And we haven't been able to distinguish that yet. So the things that we do know right now are the change in GATA3 abundance regulates somehow the initiation or emergence of D and 3 stage quiescence and allelic exclusion. So the predictions of the hypothesis that GATA3 might regulate allelic exclusion were that wild-type DN4 cells will have 50% in a productively rearranged plus DJ configuration, and 50% of the cells would have a VDJ plus, VDJ minus configuration. That was true. That in GATA3 heterozygous hypomorphic mutant mice, that GFP minus cells would have one productively and one germline configuration TCR beta transgene and TCR beta gene, and in GFP positive cells, both TCR beta alleles would be fully recombined, and that's true. Both TCR beta alleles in TCR alpha beta genes will be unrearranged in DN4 cells, and that was the experiment I alluded to a minute ago but didn't show you, and we found that that was also true. And finally, mice expressing excess GATA3 and thymocytes forcibly overcome allelic exclusion, and that's true. So what we think is that right now is that GATA3 is required for multiple stages of T cell development, and we're investigating at least the beta selection step called, which is where allelic exclusion is executed. GATA3 is regulated by at least two enhancers, we believe. We haven't proven yet. And that one regulates ETP in late T cell development that is regulated by a combination of multiple tissue-restricted and ubiquitous factors. Preliminary experiments indicate that several genes are induced by GATA3, can partially rescue mid-thymocyte development, and finally, appropriate regulation of TCR beta allelic exclusion is intimately dependent on the concentration or activity of GATA3. So the key mechanistic question remaining, and one that we are pursuing at the present time, is how does GATA3 regulate allelic exclusion? Does it do so by turning on target genes that cause this negative feedback to suppress second allele recombination, or does it regulate target genes that act downstream of allelic exclusion, leading to productive expression of only the T cells that have undergone one successful recombination event at the TCR beta locus? So I haven't mentioned them enough, but these are the people who did these experiments. Most of the early experiments were done by Tomo Hosoya and Saki Omura. More recent experiments have been done by Chaju Ku and Qingqing Wu, as well as some help from Greg Myers. These other guys, these other characters in the picture are in the Globin group, and I didn't talk about that here. We've had a lot of help with these T-cell studies, both at the University of Michigan and abroad. We have a lot of current collaborators, and we've had a tremendous amount of help from the Department of Computational Medicine and Bioinformatics. Many colleagues in that department that we have collaborated with on the various papers I've told you about. So that's it. I'd be happy to take any questions. I'll let you look at these people as much as you want. Any questions? Yes.
So I'm a physicist, so I do not completely understand your talk. I will try my best to comment some questions. So, but I want to ask the question is that since that this kind of regulation is an external organ, so does it mean that if that organ is damaged, so the whole regulation system will have problems?
Interesting question. So one of the features that I didn't tell you anything about was that as the cells mature, they actually mature in different compartments of the thymus. And each of those compartments produces a different set of cytokines and chemokines and signals to specifically activate receptors at different stages. And they're in physically different locations in the thymus. So if you damage the thymus, the possibility is that you could definitely damage the entire process of T cell development. But the thymic epithelium and the thymic medulla operate essentially independently, provide independent signals. And so you would have to do severe damage to really inhibit one specific set, one specific stage of T cell development. You could inhibit the number of cells by damaging the organ severely, but it would be difficult to affect a specific stage. Come on, ladies. I can see you all talking to each other. You must have questions. Yes?
Does GATA3 also regulate the TCR alpha chain?
I don't know. Interesting question. I don't know. TCR alpha undergoes recombination actually at a different stage of T cell development. So, once cells get outside the thymus, I'm not so interested. But yeah, it's at the very end of thymocyte development. So the stage I talked about is the stage between DN3A and 3B. That's how you make that transition, is to produce a beta protein. And it recombines with a protein called pre-TCR alpha to decide whether or not to feed back and allow or not allow second allele recombination. The real TCR alpha that allows the T cell receptor, the full T cell receptor, to be expressed on the cell surface takes place at a later stage.
Is GATA3 expression sustained after the GN3 stage?
So GATA3 is required at different levels at all of these stages. That's one of the really curious things that we don't understand yet, but it's expressed at its highest level, actually, at the DN3A stage. That was one of the clues that originally led us to investigating allelic exclusion. It's also required once T cells reach the periphery. It's required for expansion of CD4 cells and for a massive expansion of T helper cells, Th2 cells. So, its regulation is really complex, but if we just follow the levels of RNA, which is about all we can do with thymocytes because there are so few of them, it varies by more than an order of magnitude at different stages. Yes?
Any reason to think that any of the other GATA factors are doing similar things in other lineages? We haven't looked. From my perspective, I think the more interesting question
is whether or not GATA3 goes through a monoallelic to biallelic switch in any of the other tissues where it's expressed. And we are investigating that. We have no evidence for that yet in the sympathetic nervous system or in kidney development or chromatin cell development, which are the only two other tissues where we've looked. No, I don't know. I guess I don't have an opinion. It could, since erythroid cells actually go through a very nicely staged development, it could occur in erythroid cells with GATA1. But using GATA4 or GATA5 in muscle development is a lot, early development is not really as well defined. So it would be much harder to study, I think.
Are the cell lines derived from thymocytes good models to study thymocyte biology or mature T cell biology compared to primary cells?
So a lot of the work on T cell biology has been accomplished using cell lines from transformed cells. And since they express really exquisitely different cell surface markers, you can categorize those cell lines very nicely. But we primarily used mouse models because we like to see things happen in vivo. Sometimes we do use cell lines for initial tests. For example, the rescue experiments were initially performed in a T cell line that is more or less like a bone marrow early progenitor cell that we know expresses GATA3. And so we tried some of our initial retrovirus, lentivirus rescues in those cells, but we tend not to use cell lines that often. Yes?
Is it possible to study more explicitly what the actual mechanisms, sort of geometrically and topologically, that would regulate what's going on, and thereby accessing more directly what the interrelation between the mechanisms are? Yeah,
we would love to be able to do that. So everything I've described for you, except for the transcriptional activation stuff, is pretty phenomenological. And that's why I left you with the question of, what is GATA3 actually doing? The mechanism that it could be eliciting could be as simple as it's required for differential activation of the recombination activating genes that actually execute the DNA recombination, or some other gene that is directly involved with the DNA recombination mechanism. We just don't know yet. We're looking at those kinds of things, but they haven't been as productive so far. So we've taken this rescue approach to try and see if we can identify downstream targets, and hoping that that will give us some clues as to mechanism. Any other computational questions or mathematical or physics questions? All right.
Well, we can discuss during the break.
Okay.
Okay. So we have a small gift from IAS to you. So here. Thank you very much.